Hi, welcome to our lecture for today. Today we are going to be talking about cell membranes, what their structure is and how they function, and within that context, diffusion and osmosis and transport across the membranes of other substances. Okay, so just a reminder that cell membranes are composed primarily of phospholipids. Phospholipids are a type of lipid, and we discussed those in the last lecture. Phospholipids have two fatty acid tails that usually have some unsaturated unsaturated component, so some double bonds, and that makes them fluid. And they also have this head that is nonpolar and hydrophilic. Okay, the fatty acid tails are hydrophobic, so they try to get away from water, and the polar head is very hydrophilic, likes to interact with water. So this phospholipid bilayer is what cell membranes are composed of. And again, we're talking about not only the membrane that's around the entire cell, but also the membrane that is around all of the organelles. Okay? So all of these cell membranes are composed of a phospholipid bilayer that are composed of all these phospholipids that are arranged so that their hydrophilic heads or polar heads are facing either out of the cell or toward the inside of the cell, and that's because there's water everywhere. So inside the cell there's a lot of water, the cytoplasm is full of water, and then outside of the cell is also water. So if we're talking about a single-celled organism, then it's swimming around in water. If we're talking about a multicellular organism, then all of the cells are bathed in a solution of water. Okay? And of course the organelles are all surrounded by the cytoplasm, so they're full of water as well. So there's the phospholipid bilayer, and then in addition to that, there's also a bunch of proteins that are embedded in the membrane. Okay, and this creates what's called the fluid mosaic model of cell membranes. The fluid part of it comes from the fact that the phospholipids act as a fluid because they have unsaturated fatty acid tails. In addition, we have these guys, which are cholesterol, that are embedded in the membrane and help control the fluidity. And then the mosaic part means refers to the proteins being studied in the background of the phospholipids. So when you say you make a mosaic picture, you would have some kind of background, usually like maybe concrete, and then you would put pieces of glass in that. The concrete is the same as the phospholipids, and the pieces of glass are the membranes, or membrane proteins, excuse me. So these proteins are embedded in the membrane, and they're really important for the function of the membrane. So membrane proteins can add all sorts of functionalities to the membrane, in addition to the functionality that the membrane has just on its own. Okay? So there are multiple different types of membrane proteins. They are all embedded in the membrane, and that means that they have some portion of that protein molecule that's sticking into the inside of the membrane, and then some poking out on either side. Some membrane proteins are just stuck on one side and poke out to the inside or to the outside, but all the ones shown here go straight across. Okay? Keep in mind that there are thousands of different kinds of membrane proteins, and that they all fit into one of these categories or classes of membrane proteins that do specific jobs. There's definitely not like one membrane protein that does all of these. There's a ton of them. So one of the most important, and the one that we'll come back and talk more about today, are transporter or transport proteins. These move substances across the cell membrane, either from outside the cell to inside the cell, as is shown here, or opposite. Okay? So they allow substances to go across the membrane. Okay? Another category of membrane proteins are enzymes. Enzymes catalyze reactions and cause them to occur fast enough for life to exist, and we're going to be talking about enzymes in our next lecture. So suffice it to say that some of those enzymes are embedded in the membrane. There's also receptor proteins that are able to receive signals from other cells or from the external environment, and these receptor proteins kind of act as a lock, and then the signal, which can be a protein or could also be a hormone, fit into that lock, and when the signal binds to the receptor, that transmits the signal and causes some chain of events to happen inside the cell. Okay. There's also recognition proteins, and that's what's shown here. These are supposed to be some carbohydrates that are attached to it. Recognition proteins are important because they allow your cells to recognize other cells that belong to you or cells that don't. So these are really important for our immune function. And then finally, there's connection proteins. Connection proteins allow cells of the same kind of tissue to stick to each other to form that tissue. And they also allow for connections with the cell membrane and the cytoskeleton, or even with the cell membrane and the surrounding extracellular matrix. All right, so that's what the membrane is composed of, the phospholipids with the membrane studded in that, making the fluid mosaic model of cell membranes. And now we're going to talk about membrane function. Okay, remember that one of the main functions of the cell membrane is to act as a gatekeeper, and you could apply that also to the membranes that are around the organelles. Right, so the organelles are doing specific jobs and also need to regulate what goes in and out of them. So this is an important function that all cells and organelles need to do, and just the basic function of the phospholipid bilayer allows a lot of gatekeeper functions to happen. Okay, so this structure where you have it's kind of like a sandwich, right, with hydrophilic bread and hydrophobic inner layer, kind of like the peanut butter maybe, allows the cell membrane to be selectively permeable. That just means that it allows some things across while not allowing others. So it's kind of like selecting for some things and not. Okay, and it's that hydrophobic inner layer that makes it so that some things can't get across. Okay, so before we talk about that, though, we need to talk about diffusion and just get a general sense of what that is underneath our belt. It's not a hard concept to grasp. Diffusion is simply the process by which substances move from a high concentration, like where these dye molecules have just been dropped in, to a lower concentration. Okay, so if you put in a drop of food coloring into a glass of water, those dye molecules are going to move out, and they keep moving until they're completely spread out, okay? Until equilibrium is reached. This happens simply because the molecules are in constant motion, so they're constantly banging into each other and moving each other around. Okay? You could cause diffusion to happen more quickly if you heat up the liquid or the air, for example, if something's diffusing through the air. So when you make tea, you use hot water to make the tea molecules diffuse faster. You can make tea with cold water, but it would take a lot longer. Other than speeding it up, you don't need to put in any energy. So no energy input is needed. That means diffusion is just going to happen on its own. It's a natural process. Things will tend to move from high concentration to low concentration. Okay. So when we're talking about diffusion, we also need to understand concentration gradients. Concentration gradient is just this. When you have more of a substance and less of a substance over here, and then things will tend to go from high concentration or low to low concentration. We call this diffusing down the concentration gradient, or moving from high concentration to low concentration. Okay? Substances will tend to diffuse down their concentration gradient until equilibrium is reached. Okay? Equilibrium is when things are completely mixed, okay, and there's no more concentration gradient. One thing to keep in mind is that the molecules are still moving around and banging into each other. There's just no additional mixing to occur because they're fully mixed. All right, so now we're going to talk about this. Now that we know what diffusion is, movement of substance from high concentration to low concentration, we're now going to talk about it in the context of having a cell membrane. Okay, so if you have a cell membrane, that's going to block the movement of some things that maybe would want to go down their concentration gradient. Okay, so the idea is like this: inside the cell, you have a bunch of solutes, so you have a bunch of things inside the cell, substances, and outside you have some too, but less. Right, so we're talking about diffusion across a cell membrane from high to low concentration. Okay. So like I said, the hydrophobic inner layer allows the cell membrane to be selectively permeable, and it makes it so that some things can go across and some things can't. Okay, things that can just go right across the cell membrane are hydrophobic molecules because they have no trouble going across that hydrophobic inner layer, right? They're not repelled by it at all, okay? Also, small uncharged polar molecules, and really we're just talking about water when we're talking about this, can also go through, okay? Water is a very tiny molecule, and so basically it just squeezes its way through, even though it doesn't like that hydrophobic layer. It can just get through. And then there are some things that just can't get through because they are polar, large and polar. Things like sugars, amino acids, nucleic acids. A lot of the things that are dissolved inside cells cannot go across the cell membrane. And the same is true of anything that has a charge, a full charge, an ion. Okay? Because of that hydrophobic inner layer, these molecules or substances are unable to go across. Okay, so, but the cell does need to regulate what goes across the cell membrane, and oftentimes needs to bring things that wouldn't cross on their own into or out of the cell. Okay, so now we're going to talk about how do substances cross cell membranes. Okay, there are two basic ways. There's passive transport and active transport. Right now we're just going to address passive transport, and then at the end of the lecture we'll come back and talk about active transport. So passive transport are for things that are diffusing down their concentration gradient. So they're going from high to low, and remember that's a situation that will just happen naturally, so you don't need any energy input into the system. Within passive transport, then there are two categories or types of passive transport. Okay? The first is simple diffusion. Simple diffusion is for those molecules or substances that can go across the cell membrane on their own without any need for a helper. Okay, the primary ones that we will kind of be talking about this quarter are oxygen and carbon dioxide. Remember, oxygen needs to go from your blood into your cells so that your cells can keep doing cellular respiration, and carbon dioxide needs to do the opposite. Water can also go straight across by simple diffusion. And actually water that's diffusing is called osmosis, or diffuses by the process of osmosis. And so we will talk, because water is so important because it's everywhere, we're going to talk about water separately in how osmosis works. We're going to spend some time talking about that in a few minutes. Okay, so that's simple diffusion, and here's the example using oxygen. Okay, so oxygen often, say you're running along and you're breathing and you're sending oxygen down to your muscle cells, right? The muscle cells are using up the oxygen in the process of cellular respiration, and so they're becoming depleted of oxygen. Your blood brings fresh oxygen from your lungs to your muscle cells, okay? And so this would be like the blood, and this would be inside the cell. Because there's more oxygen in the blood than in the cell, the oxygen will move by simple diffusion from outside the cell into the cell, okay, just going down its concentration gradient. Carbon dioxide would be doing the opposite. Okay, then there's also facilitated diffusion. These are for things that they're still going down their concentration gradient and it doesn't require energy, but these are things that can't go across on their own, right? So they're repelled by that hydrophobic inner layer, so they need some help. So facilitated diffusion, facilitated means helped, okay? So it's still diffusion, just going down its concentration gradient, but it's helped, and it requires a transport protein. Okay? So here are two examples of substances that would not be able to cross the cell membrane on their own. Glucose is polar, and chloride ions are charged, right? So they're going to be repelled by that hydrophobic inner layer, and yet they're moving from where there's more, so a high concentration, to where there's less, a low concentration. Okay, so they can still move down their concentration gradient, they just simply need some kind of helper. Okay, and that is provided by a type of transport protein called a carrier protein. The carrier protein has a binding site that's specific for whatever substance it's moving. So for this one, it'd be a glucose carrier protein, and simply allows that substance to move down its concentration gradient. Okay, doesn't take any energy to do this; it just needs a helper protein. Okay, and then the other one is the chlorine. Okay, the chlorine also has a high concentration, in this case outside of the cell, and this is low inside the cell. by the way, you could have these reversed. So cells are not always bringing things into themselves. They're frequently taking things out as well, exporting things via facilitated diffusion. So you could get the opposite direction as well. Things will just move down their concentration gradient. So for the chloride ion here, it's charged, and so what it needs is some way to get across. Okay? And usually this is often provided by a channel protein that will just allow the chloride ions to go down their concentration gradient, and these are highly specific too. Your book talks about aquaporins. Aquaporins are for facilitated diffusion for water. So water can diffuse across the cell membrane by osmosis, but sometimes that's not enough, and so there's also this facilitated diffusion that can happen. All right, so let's talk about osmosis. So osmosis is simply a special case of diffusion that's talking specifically about water molecules that are diffusing across a selectively permeable membrane or a cell membrane. Okay? So the important thing to remember here is what you just learned about diffusion, that substances will move down their concentration gradient, is the same for water. We're just talking about water molecules moving down their concentration gradient from high to low concentration. But the idea is the same. Okay? We care about this because water is everywhere inside of cells and outside of cells, and cells really need to be careful to keep their amount of water, called their osmotic balance, in check so that they don't become too dilute or they don't become dehydrated. Okay? And remember that the cytoplasm has a bunch of things dissolved in it, right? I remember I said it was a jelly-like substance with lots of stuff dissolved into it. Those things that are dissolved in it are solutes. And the question we are always asking is, which way will the water move by osmosis? Okay? In other words, will it move into the cell, or will it move out of the cell, or will it do neither? Will it kind of stay the same? Okay? And in order to have a discussion about this, we need to make sure that we all understand what a solution is. Okay, so we're always talking about solutions and the concentration of solutions. So a solution is simply a solute plus a solvent. When we're talking about biology, we're always talking about water being the solvent. Okay, and remember, water can dissolve things that are polar, that can hydrogen bonds, or ionic ions that can also make hydrogen bonds. Okay, so the solutes have to be something like that: sugars, salts, amino acids, that kind of thing. Okay, you put those together and you get a solution. Okay, so we could have our imaginary solution here in a beaker. Okay, and when you make a solution, the amount of, I'm just going to erase that because it's in the way, the amount of solute to the amount of solvent gives you the concentration, or tells you how strong it is. So we're just going to look at a couple examples here. So for example, you could have a 3% sodium chloride solution. Okay. What that is telling you is that 3% of that solution is made of sodium chloride atoms, and 97% is water. Okay, so it usually doesn't tell you that it's a 97% water solution. It'll tell you it's a 3% sodium chloride solution, and you have to fill in that the rest is 97%. Okay, so of course, just in case, I have to spell it out. So, you know, the whole solution will be 100%, and if 3% is sodium chloride, then the rest will be water. Right? So, similar kind of thing. If you have a 10% glucose solution, that will mean that 10% of that solution is glucose and 90% is water. Okay. Why am I going into this? This seems obvious, right? Well, when we start trying to answer these questions, which way is the water moving, being able to figure out what the water concentration is really important. Okay. I will also like to point out that it's just the total solute concentration that matters. So inside the cell, there's a whole bunch of solutes that are dissolved in there, and it's their additive effect to their total amount of solute that matters. So for example, we could have a solution that was both 3% sodium chloride and 10% glucose, and that would mean that the total solute concentration was 13%. Okay? So that means 13% of that solution is some kind of solute. It actually doesn't matter what kind it is. It can be a total mix of different ones. it is the total solute concentration that matters. Okay, so if it's 13% total solutes, then it's just 87% water. All right, so let's set up a little scenario, and you're going to be doing something similar in lab this week. So we're going to set up a scenario where we have this beaker with a solution in it. I'll be the surrounding solution, and then we're going to have an imaginary cell. Okay, we're going to do like a model cell in lab this week, where we're going to make a cell out of cellulose tubing that has tiny little holes in it that will like be a model for a cell membrane. Okay, in this case, we're imagining that this is a real cell that has a cell membrane around it. Okay, and the thing to remember is that only water molecules can move into the cell or out of the cell, and the solute is stuck on whichever side it started with. Okay, so only the water can move down its concentration gradient, not the solute. All right, so let's look at a very basic example where we have a solute concentration of the solution surrounding the cell of 1% and the same inside the cell. So 1% for each of those is solute, and that would mean that 99% is water, right? So we're trying to answer this question, which way will the water move? Okay, so the thing to think about is that water is going to move down its concentration gradient. Well, in this case, because we have equal, that means that the solute concentration inside and outside of the cell is equal, as is the water concentration, right? So it's 99% either way. Okay? That means that we're going to see no net movement of water molecules, okay? And this is called isotonic. Okay? It doesn't mean no water is moving across the cell membrane at all, because remember, the water molecules are bouncing around, and some will go across, okay? But the same is happening inside the cell, and an equal amount will go out. Okay? So this is equilibrium, right? Equal amount on either side. Okay? That's isotonic. Okay, here's a different situation. Here we have changed the solute concentration of the solution to 3%. And kept the cell the same at 1%. And just remember that only the water molecules can move. So the solute, whatever it is, and it could be a combination of anything, the solute stuck, okay? So it can't move, and just the solvent can move, just the water. Okay, so here we have a 3% solute concentration on the outside of the cell and stellar 1% on the inside. Now we've changed our relative amounts, right, from outside versus inside. Now we have on the outside a solvent concentration or a water concentration of 97%. Okay? So now we've set this up so that the solute concentration outside the cell is greater than inside the cell. And then conversely, the water concentration is less on the outside than on the inside. Okay? And remember, we're still trying to answer this question. If this was a real cell, what would be happening? Would water be moving into the cell or out of the cell, or neither? Okay? And the way to answer this is to think about the water concentration. And I really like this, the thinking about the water concentration, because thinking about just the solute can sometimes very much confuse you. And if you just come back to the basics of what is the water concentration, then you can figure it out. Okay? So if water is going to move down its concentration gradient from where it's high to where it's lower. So in that case, it's higher inside the cell, and water will move from the inside to the outside. Okay? So water will move to the from the inside to the outside. Okay, I said it right. Okay. So lots of water will move from the inside to the outside. We will still get some movement of water from the outside to the inside, just from the random movement of water molecules, but the net movement will be from the inside to the outside. Okay, that's what that says, and this is called a hypertonic solution. I would just like to pause here and tell you guys that I am not super concerned that you remember this term, isotonic, hypertonic, or the other one, hypotonic. What I am concerned about is that you can predict which way the water is going, okay? And you don't even need to remember the word hypertonic to figure that out. You just have to remember that water will always go from high concentration to low concentration, okay? So if we have higher on the inside, and that would mean less solute, then it's going to move to the outside. Okay, let's look at the opposite. Here I've changed it to a 0.5% solute concentration on the outside and kept our cell at 1%. Okay? That means that on the outside we now have 99.5% water and it's still 99% in the inside. Okay, so the solute concentration on the outside of the cell is now less, which means the water concentration will be more. Okay, so which way will water move? It's going to go from where it's more to where it's less. Right, and water will tend to move from the outside to the inside of the cell, the opposite. Okay, and this is called hypotonic. And again, not super concerned about you memorizing that. True, if you're going to go on to anatomy and physiology and microbiology, then they will probably require that you know those. I am mostly concerned about, can you solve this problem? Which way will the water flow? Okay, so this will be net movement into the cell. All right, so very well and good. And here's a diagram that just shows all that same stuff. Okay, going either, this would be isotonic because it goes both ways and it's equal. This is hypertonic, more solute on the outside, water goes out. And hypotonic, water goes in. Okay? When you're thinking about the cell, then the size of the cell is going to either shrink or expand based on, or stay the same based on what water is doing. So if water is being pulled out, then that will dehydrate the cell and it'll shrink it. Okay? If water is going in, it'll expand it and make it larger. And if water is not going either way, no net water change, then it would stay the same size. So this is really pertinent to thinking about cells. Okay, so we're going to look to see, this summarizes what happens to animal cells and plant cells in those three types of solutions. Okay, so let's take hypertonic first. Okay, hypertonic solution is when there's more solute on the outside of the cell than on the inside of the cell. Or another way to think about that would be less water on the outside than on the inside. Okay, so water will tend to move from the inside of the cell to the outside, and the cell will lose water. Okay, so we're just looking at the hypertonic here. We're ignoring the stuff over here. Animal cells, remember, have a cell membrane around them, right? And then they have the cytoplasm that's full of water. If you put an animal cell in a hypertonic solution, say if you take a red blood cell that has about 1% solute concentration, I think it's more like 0.85%, but about 1%, and say you put it in 3% sodium chloride, what's going to happen is that water is going to move out of the cell, right, by osmosis, and that will shrivel the red blood cell and kill it, right? So it'd be really bad to give someone as an IV a 3% sodium chloride drip, right, because that would be hypertonic. It's also bad news for plant cells to be in a hypertonic solution. Plant cells have the cell wall around them, and the cell wall is actually completely unaffected, unaffected by the solute concentration. Water and solutes can go right across it. But remember, plant cells have that large central vacuole that's full of water, and then their cytoplasm is full of water. So if you put a plant cell in a hypertonic solution, then it's going to suck the water out of that, and what you will see, and you guys are going to see this in lab this week, you will still be able to see the cell, you will still be able to see cell wall. It'll be unaffected, but you'll see this plasmal, it'll become plasmalized. The cell membrane will pull apart and everything will shrink. Okay, so that's what happens. So hypertonic is bad news for animal cells, plant cells, bacterial cells, fungal cells, and that's really good for us because we take advantage of this to preserve our foods. Okay, so jams and jellies are made by taking the fruit and adding a ton of sugar. Well, the sugar acts as a solute, right? It's dissolved in the water, and it is extremely hypertonic. So if you have some jam or jelly and some fungal spores or some bacteria land in it, that they're going to dehydrate. They're going to get all the water pulled out of them and they'll die. Okay. Same is true of honey. Honey never goes bad, and that's because it has a 20% solute concentration. Really, really, really hypertonic. Okay. So if a fungus spore or a bacteria or something lands in the honey, it's just going to get shriveled up and die and won't grow into that substance. Okay. And then we also use it to preserve meat. So jerky uses this. You take the meat and you put a whole bunch of salt on it, right? That makes it super hypertonic and makes it so that anything that lands on it will not survive. All right, now let's talk about isotonic. Okay, isotonic is when it's equivalent, right? And so there's some water movement going both ways, but no net change. Okay, this for animal cells is a super happy situation. Okay, that means that they have the amount of water that they need, and not too much and not too little. So they're not shriveling and they're not doing what the other thing is, which is tend to explode. Okay, so people, if you become dehydrated and you go to the hospital and get an IV drip of saline, that's just a weak solution of saline, which is salt water. It's about 1% saline solution, which is isotonic to your blood. Okay, and that bathes your blood cells in a nice solution where they can have osmotic balance. Okay, isotonic solution is not like super great for a plant, and I'll explain that in a moment why, but it doesn't really super harm it either. But isotonic is really super happy for animal cells. Okay, so let's talk about what happens with in a hypertonic solution. Maybe, sorry. Okay, excuse me. Let's talk about what happens in a hypotonic solution. Okay, so in a hypotonic solution, the inside of the cell has less water in it than the outside, and water will tend to move from the outside in. Okay, if you put an animal cell in that, like a red blood cell, what's going to happen is that water will keep going in and going in and going into the cell, and eventually, because it just has the cell membrane, it will eventually burst. Okay, so that would be very bad. That would be like if you put pure water in an IV drip and gave it to somebody. That would burst all of their red blood cells and could actually kill somebody. So that's, that would be bad news. But for animal or plant cells, a hypotonic solution is the best for it. Okay. And that is because of that action of the central vacuole and the cell wall working together to make that sturdy structure. Okay. So remember, in order for a plant cell to be sturdy and be able to stand up against gravity, it has to be turgid, which means that its central vacuole has to be full of water, and the water inside the central vacuole will push out against the cell wall, and that will create turgor pressure. Okay. In a hypotonic solution, water will be constantly flowing into the plant cell. Okay. But because it has that cell wall, the cell wall pushes back on the cell membrane, making it so that it can't overfill. So plant cells won't burst, whereas animal cells do. Plant cells don't because they have that cell wall. Instead, they stay nice and plump and happy. Okay? And that makes a very happy plant that is fully hydrated and has turgor pressure and can stand up against gravity. So, if you forget to water your plant, then you're putting your plant cells in a hypertonic solution. Or if you give them too much fertilizer, too much solute, that can happen to them also, and that would dehydrate their cells. Right? Or if you have some, say, celery that's in the refrigerator and you've let it dry out, it's become dehydrated, you can plump it back up by putting it into a hypotonic solution, because then water will flow in. All right, so that is isotonic, hypertonic, and hypotonic. And remember, I really don't care if you memorize those. I'm not going to put on a test, define this. I will ask you to do some problem solving. Here's a cell in the solution. What's going to happen? That kind of thing. All right, now finally, let's get back to how substances cross the membranes. Remember, we said there are two ways. We talked about passive transport. That's diffusion of substance down its concentration gradient, and it doesn't require energy. But cells oftentimes need to move things from where there's a little of it to where there's already a lot of it. Say, for example, a mitochondria that's doing cellular respiration. It will constantly need a flow of materials into it where it already has a lot of it. Okay? And this is called active transport. Okay? It's active transport because it's moving a substance. I shouldn't say diffusion. Actually, I should say movement. I should change that. It's not diffusion, it's movement. So it's movement of a substance up its concentration gradient. Okay, this is the opposite of what it wants to do. It'd be like you pushing a car up a hill, right? It's going to take a lot of energy, and so this requires an energy input. You know, if you have a car at the top of the hill, you don't have any brakes on it, you let it go, it's going to go down on its own, right? That's just going to happen. But if you want to get it back up to the top of the hill, then you need to push it up and you need to put in some energy. Okay, so it requires an energy input, and it also requires a membrane transport protein because things aren't going to just simply diffuse down their concentration gradient. They're doing the opposite. Okay, so that's what this is showing here, right? So here's the cell membrane, here's a transport protein, and it's requiring energy. Energy can be provided by the molecule ATP, or there's other sources of energy as well. That's one of the things ATP is used for. Okay, and as you can see from here, there's more of the solute inside the cell than outside the cell, so it's going to be pressed, or it's going to be had active transport